Okay, can everyone hear me, I guess? I have a bit of a cold, so if I'm not understandable, then let me know. Well, first of all, thank you very much for inviting me. It's been a really interesting conference so far. So I'm suffering a bit from imposter syndrome because I'm not really from developmental psychology. I'm more of a cognitive neuroscientist studying perception and perceptual decision making. But for me, up to now, it has been very interesting. So I hope that, that this kind of step away from developmental neuroscience for you will also be interesting. So what I'm going to talk about today is sort of my research theme, which is about like how do we make perceptual decisions, focusing on a very sort of active and predictive process, such that we're not just registering the environment and making a decision based on what we accumulate or what we get in, but that this is a very active process of testing hypotheses against all the knowledge and expectations that we already bring to bear on this problem. And to quickly illustrate this, if you've never seen this image before, you might have no idea what you're looking at, so, because this is a so-called Mooney image. If I now give you one perceptual experience, and I move back to the original, you can see that it has really transformed now, right? I mean, whereas you probably had no idea what you were looking at before, from this point on in your life, you will always be able to recognize this very quickly. Now, I always find this very impressive. Just one experience in your life, in a way, can change perception in a very enduring way. And this illustrates to me that past experience and knowledge is hugely important in even simple processes like perception. So here's another example. When we look at this in isolation, it's, again, very hard to see what this is. If we now take this same stimulus, but put it in two different spatial contexts, you can see that the same image, so it's exactly the same image except for a 90-degree rotation, and it has taken on a different meaning just by virtue of the context in which it's embedded. And here again what this shows is that the brain may be building up hypotheses about individual elements based on the context, in this case a spatial context that dictates that certain objects like cars tend to be at certain locations within a scene. So how can we conceptualize these kind of phenomena, these kind of context dependencies? So within my lab, we have been thinking about this within the framework of what you could call the hypothesis generating brain. And as I just showed you, the outside world is what you could call hierarchically organized, where you have a scene which is composed of objects composed of even smaller objects, etc. And you can see also in this particular example that this car is more easily recognized within the context of a scene, just like in a previous example, but also individual elements like over here, the tail light is not identifiable on its own, but becomes very easy to identify within the context of the car. So the brain sort of mirrors this principle. So you have also neurons in the brain that are dedicated to the analysis of individual elements in a scene, but also to more complex conjunctions of elements, all the way up to scenes. And one way of thinking about, for example, a contextual influence here is that perhaps feedback from higher order regions that are encoding things like objects or scenes constrain the processing of these earlier neurons that are dedicated to the analysis of simple features. All right, so there's two key ingredients of what I would call the predictive brain that I would like to walk you through today. The first one is the idea that the brain is continuously generating predictions about causes in the world, for example, based on spatial and temporal statistical regularities. And one potential neural implementation of that could be that feedback projections generate what I call virtual data, and I will get back to what I mean with that in a second. And those predictions then, or those virtual data, are then updated by the incoming sensory data, potentially by forward projections that update these predictions. So now let me go to the outline of my talk. So the first question I'm going to try to answer is there actually empirical evidence for the notion that the brain is generating virtual sensory data? And with virtual sensory data, I mean that it's not that the brain is creating a visual, for example, or perceptual experience without there actually being any evidence for that experience yet, but which then can be sort of tested again or can serve as a template for the incoming input. So when we anticipate a particular perceptual event, we might already generate the associated sensory activity so that then can be quickly, or that can bias or be confirmed by the actual incoming data. So that's the first question I'm going to tackle. Then, having shown that that is actually the case, I'm going to ask the question, why don't we then hallucinate such virtual sensory data? So if we are creating sensory events, how can we keep those apart from the actual data that is provided by the eyes, for example? And then in my last part, I will briefly touch upon how do these virtual data or how do these sensory predictions actually interact with reality? What is the interplay between our predictions and the incoming sensory information? So let's start with the first one. So does the brain generate virtual sensory data? And one scenario in which it might be a good idea to generate virtual sensory data is when you are playing a tennis match against Roger Federer. Because as you all know, he's a world-class tennis player, and but even though, so he's very quick, but he's not defying the laws of the universe. So in that sense, his actions and the consequences of his actions have a certain predictability to them. In the sense that objects are typically static, but if they move, they also move in a predictable way. So when you are in a situation like this, you might predict that the ball at particular time points in the future will be at a particular location. So that is a very simple form of predicting the future, which might be helpful in this particular example. Now we know that in the brain of rats, but also there are cells, for example, in the hippocampus, but also in the visual system, that can sort of forward replay, and in anticipation of an actual event, they can forward replay that particular sequence. So for example, when a rat has been exposed to a certain environment and has walked a particular trajectory, then at the beginning, when he is placed at the beginning of such a trajectory, you sometimes see spontaneous waves of activity that sort of replay or forward replay what the rat is likely going to do next. Now that has been observed in the hippocampus, but it has also been observed in the visual system, when people, or in this case again rats, when they're exposed to a sequence of visual events. Now we were curious about whether such forward replay, which is in a way sort of like an anticipatory wave of activity, replaying or pre-playing, maybe more aptly said, what a certain individual is anticipating. For example, in this tennis match. So what we did is we had people repeatedly observe moving dot sequences, a little bit like a tennis ball that would move from the left to the right on the screen. And then every now and then, after having been exposed to that repeatedly, we had a situation where we would just flash the starting point, and we were curious to see whether that would trigger sort of a wave of anticipatory activity at the corresponding sort of neurons that represent that location and also at the corresponding time. And we're doing this in the visual cortex because we know the visual cortex has a very nice map of visual space, a retinotopic coordinate frame, so we can actually measure at high precision whether the right what is represented where. So let me explain you that in a little bit more detail. So what we do here is or what we use here is a technique called population receptive field mapping. And that sounds very complicated, but it's actually, it's not that bad. What it means is that for every sort of little voxel in the human brain that we measure in the visual cortex, we establish what location it is most sensitive to, so what it reacts to most, and also how large that the receptive field is, so how large the area is that it responds to. And when we know that, so when we have that, then we can actually present a sequence of dots and then see whether there is activity in the correct sort of visual field locations. So here I'm showing you the result for the main task, where there is like a dot sequence from left to right. And here in dotted in black is the actual locations where the stimulus was presented. And then in yellow, sort of overlaid in this color map, is the fMRI bold activity as measured for all of visual cortex, but now sort of transformed back into the stimulus space. So it's not in brain space where it would be hard to interpret it, but it's already transformed into stimulus space, such as you can see that there is indeed activity exactly at the locations where the brain was stimulated. So this is of course just a sanity check. This is just to show that we can actually, with high spatial precision, reconstruct what people are looking at. So now we're coming to the more exciting part, which is what happens when we are just showing this first dot after having paired it, or after people are starting to expect to see a tennis ball moving from the left to the right. So here, this is what I already showed you here. Now is this is when we just flash the starting point, so our pre-play condition. And this, for comparison, is when we flash the endpoint. So when there is no prediction being made about the ball moving, because the ball moves always from the left to the right. And you can see there's a marked difference between those two activity maps. So indeed, when we just flash the starting point, you can see that there is activity at all of these other locations, although it is, of course, gradually falling off. So that suggests indeed that there is, at least in terms of the spatial activity, or spatial map of activity, there is an activity just triggered by the anticipation of perceptual events. Now we can do something a little bit more exciting here, because we measure the neural activity here with very quick temporal resolution. So what we can do is actually look at whether the cascade of, or whether the activity in the human brain also follows the temporal order. So again, just as a sanity check, I'm first showing it to you when you flash the sequence, so when you have like left, all these dots one after the other. And what you can see is that, it's actually not very well visible perhaps. Well, there is a delay, because that's because of the bolt signal, but then you see that there is basically these hills of activity from starting with the most left location and then moving on to the right. So even though the bolt signal is very slow, it's very sluggish, there is actually quite a good preservation of temporal information. So you can also interrogate the timing of events using fMRI actually. So now what happens for the flash? So now you see these hills of activity, and you might have been able to appreciate that here again the temporal order is again preserved. So there is like a prediction of perceptual events, first at the second dot, then the third dot, and then the fourth dot, and that temporal order is preserved. And again, if we would just flash the endpoint, you see there is this kind of lonely activity at the location where you stimulate, but it doesn't progress through the visual field. Okay, well, finally, to one direction that we're following this work up now, this is still unpublished work, is even though you might be predicting this particular sequence when you see Roger Federer in this situation, he might every now and then try to trick you and do like a little drop shot on the other side. So in that sense, we typically may not actually hold one prediction about what is going to happen, but we might actually have multiple competing hypotheses about what is going to happen next. And these might happen with different probabilities. So even though this one is maybe the likely sequence, you may also have situations where you have a less, both a likely and a less likely sequence. So again, we try to mimic this situation in the lab by having one stimulus sequence being expected, and the expectancy here was created by tone. So a high tone, for example, was associated with a sequence that would go upward, whereas a low tone was associated with a sequence that would go downward. And now in 20% of the cases, this unexpected sequence would occur. And what you see here is that, that when we just show the initial starting point in conjunction with a tone that would predict the upward sequence, you see that also the pre-play is particularly dominant in the upward direction and not so much in the downward direction. Whereas if we have a neutral context in which the tones do not predict what is going to happen, you see that there is no distinction between the two trajectories. So now you have, if you will, pre-play of both trajectories. Okay, so that's the first part. And I think the take-home message from this part is that the brain can actually generate experiences when they, even when they don't happen. So just in anticipation of a particular event, the brain might already activate a certain sensory representation. But that might immediately probe the question, so why don't we then see those events, right? I mean, we may anticipate things, but we typically don't see them unless we suffer from hallucinations. So why don't we typically hallucinate these virtual sensory data? And one of the answers to this might be given by the anatomy of the cortex. So for many of you this might be familiar, but let me just very quickly review the anatomy. So if we have, this is our brain, we know that the brain is composed of several layers, actually 6 layers, but for simplicity I've now carved them up into like 3 compartments, superficial, middle, and deep. And we know that input from the eye, so all our sensory information, actually comes in the middle layer, or mostly in the middle layer, a little bit in the deep. And from there, it activates the whole cortical column and then is fed forward again to the next cortical area, again arriving in the middle layer. Whereas feedback really tends to avoid that middle layer. So feedback signals, which may embody basically all our thoughts, our attentional modulations, all these kind of cognitive modulations, if you will, they tend to avoid this layer. So they originate typically from the deep layers, and they target both the deep and the superficial layer of a preceding cortical area. So that's a very nice way, actually, that we can then try to disentangle feed-forward and feedback modulations. And this might also be a way in which thoughts can be separated from actual input. So within the context of this predictive brain hypothesis, you could then say that feedback signals may embody this generative model, whereas feed-forward signals then potentially embody the updates to that model. So your preconceptions might be living in the deep and superficial layers, whereas they then updated by information that arrives in the middle layer. Okay, so that was basically our hypothesis. But testing that hypothesis is quite challenging, especially in humans. Because you may know that, so this is a cortical column, but that is typically between 2 and 2.5 millimeters thick. So that's about the size of like 3 credit cards stacked on top of each other. So that's really quite small. So each of these compartments is about 0.7 to 0.8 millimeters, so about 1 credit card thickness. And usually when we think of fMRI as having a very good spatial resolution, that typically means 2 to 3 millimeters. So it encompasses the whole cortical column. So for this project, we had to go to much higher resolution in humans by virtue of like going to high fields and then measuring very small voxels only in the visual system. Now, what the paradigm that we used here to look at thoughts or to look at like internally generated stimuli is a paradigm that was pioneered by Frank Tong's lab while he was at Rochester. And what he showed is that there is stimulus-specific activity during visual working memory in early visual cortex. So what he did, I'll just briefly explain the paradigm because we copied it, so it will be useful for later. What he did is he presented subjects with 2 gratings of different orientation, of orthogonal orientation, and then he told them which of those to remember. So you get sample 1, sample 2, and a cue that tells people, okay, for example, in this case, you have to remember this stimulus. And then they were looking at a blank screen for 11 seconds, and then they had to do some tests in order to make sure that they were doing the task. And what they were interested in is the activity in this retention period. So the activity in the visual system while they were basically fixating a gray screen and there was no bottom-up input. And the interesting observation in this study is that in that interval, you can actually quite accurately read out what a subject is keeping in working memory. So what he is sort of mentally imagining, if you will, while there's no input on the screen. All right, so we used exactly that paradigm while measuring at very high resolution in 7 Tesla. And we used a slightly simpler analysis method than what Frank Tong's lab used for pragmatic reasons. So what we did is we separated voxels into voxels that liked the orientation that was kept in working memory and voxels that liked the opposite orientation, so that the voxels that preferred the orientation that was not held in working memory. So this is the start of a trial. So first you have a bold signal just elicited by the 2 stimuli that you presented on the screen. But then there is this intermediate interval here where people are imagining a particular stimulus. And as you can see, the overall signal drops quite significantly, only then to rise again when you have your probe stimulus over here. So, but in this intermediate interval, you can still see that there is actually a big difference in activity between the voxels that prefer the stimulus that people keep in working memory and the one that they don't keep in working memory. So this is true for V1, it's also true for V2 and for V3. So basically in your visual system, there then is this internal representation. And then, of course, the interesting question is, okay, which layers are actually activated by that internal representation? So here, what I'm plotting you here now on the right is the difference between the preferred and the non-preferred, so the stimulus-specific activity in this delay period for those 3 different regions. And what you can see is that particularly in area V1, so this first cortical area in in which visual information enters from your eye, there is no difference in activity in this middle layer, indeed, but it's only in the deep and in the superficial layers there is an activity difference. Again, very much in line with this idea that these feedback signals are only targeting those layers, the deep and the superficial. Whereas in V2 and V3, you actually see that the whole cortical column is activated. Maybe because V1 neurons, again, propagate up their belief, up the cortical hierarchy. And interestingly, this is also in line with what has been found in related paradigms in monkeys, so where they also see working memory modulations in the superficial and deep layers, but not so much in the middle layer. So that then might be the reason why we can have beliefs but not start to see those beliefs. And maybe if there's a disbalance, or if that separation gets blurred in one way or another, you start experiencing things like hallucinations, such as seen in schizophrenia. All right, in my last part then, I will briefly talk about how these sensory prediction signals are then actually interacting with reality. So how do they then indeed bias the sensory representations when stimulated by actual stimuli? One of the themes in my lab has been the notion that prediction signals may sharpen sensory representations in the primary visual cortex. So we've shown both with fMRI and MEG recordings that when you have a stimulus that you expected, that this is typically associated with a reduction of neural activity, but it is also associated with an improved readout of that stimulus. So a little bit like if you would have a picture, that you would be able to make it more sharper, if you will, if you can increase the signal-to-noise ratio of your image by having a particular prior belief about it. So this is at the level of the sensory representation, so the population activity elicited by that stimulus. But another interesting way in which predictions may be able to change sensory processing is not so much in terms of like the spatial representation of activity, but more like how the temporal duration of that activity. And we took inspiration here from a study by Carl Olsen's lab that showed that if you familiarize people with images, so if you have images, and this is in macaque monkeys. So what they did here is they had certain stimuli that monkeys viewed over and over and over again. And when they then compared the activity elicited by these familiar images to activity elicited by novel images, they saw something actually quite surprising. And what they did here, this is quite crucial, is they presented these stimuli very, very rapidly, so 120 milliseconds per stimulus. And what you can see for familiar input is that the activity rises for of course, after you show the stimulus, but then very quickly also decays again, such that it again can rise and decay, etc. over time. So it is actually able to keep up with these very fast representation rates. Whereas for novel images, you can see that it produces a vigorous response for the first stimulus, but then you don't really see much response anymore for the next stimuli. So in other words, what happens, or what we think that familiarity might do here for you is that that you're actually able to very quickly represent stimuli and then also reduce that representation so that you're ready to see new events. And we were curious whether expectations or predictions about what is going to happen next might have a similar effect on the brain. So what we did is we created familiar and unfamiliar input for our human participants, and we also created expected and unexpected input. And so let me tell you how we did that. So we had here sequences of 6 stimuli. So on each trial, we would present this kind of rapid stream of images. And we had 2 sets of images that we familiarized the subjects with. But crucially, one of those sets always occurred in a certain order. So it could start with any of these images, but then, if it started with the car, then you would get this thing, then this thing, etc. Whereas the other set did not have a particular order. So there, they became equally familiar with the stimuli, but there was no particular order in which they were learned. And then during the testing phase, so this learning phase was about 1 hour phase, and then there was a testing phase in which, of course, the stimuli that did not have a sequence did not have a particular expectation about their order, but particularly for this one, where there was a sequence, we could then either present them in the sequence or we could violate that and create an unexpected trial. And then also we had some novel trials where people basically, just to be able to make this comparison also of novel versus familiar input. So this is an example trial. So if you blinked, you might not have even seen it. So let me try that again. So if you just fixate, you can see this is PowerPoint, so probably the timing isn't accurate. But just to give you a flavor, that this is really a rather quick succession of images. And if you see this enough, you really start also indeed experiencing these elements more, more consciously, if you will. So first of all, we replicate sort of the findings that they saw in monkeys. We did this in MEG, so we wanted to have the temporal resolution. And what you can see here is within the visual system, this is corresponding to the lateral occipital complex. You see that there is activity, that there's more activity for novel than for familiar images. But you can also see that these peak-trough excursions kind of larger for familiar than for novel. And you can quantify that also by quantifying the power at the stimulus frequency. And then you see that for familiar, there is like a higher power at the stimulus frequency than for a novel input. And interestingly, we see the same phenomenon for expected versus unexpected input. So here again, so there is like a reduction of activity for expected compared to unexpected input, as we've seen many times before. But you also see that here the peak-trough excursion seems to be larger than for unexpected input. And this effect was more subtle, so it actually didn't reach statistical significance. But so we can't make any conclusive claims about this. But the topography, does seem suggestive of a similar effect of expectation as of familiarity. All right, so let me wrap up. So in the first part of my talk, I tried to convince you that the brain is continuously anticipating future input and creating virtual input based on these prior expectations. Then that virtual input seems to be encapsulated from what you could call real input by virtue of the laminar organization of the cortex. So as I showed you, there are like different layers within each area, and the layers activated by our beliefs seem to be distinct from the layers activated by bottom-up information. And then finally, in the title of my talk is How is Visual Perception Biased? But maybe a better title would be like How is Visual Perception Enhanced? Or how is it helped by the knowledge and the beliefs that we have? So I think that in the end, biasing has a very negative connotation. But I think what all these beliefs actually do is to lead to a spatial and temporal sharpening of sensory signals. And that then allows for more quick and unambiguous perceptual decisions. All right, with that I would like to thank the people that did this work, so particularly Sam Lawrence, Pim Mossert, Maria Manahova, and Matthias Ekman. The funding agencies that helped. Thank you.
Questions?
Hi, thanks for this beautiful talk. I've really enjoyed it. I have a very basic question, I think. So one prerequisite for this anticipatory process is having at least one experience. So what's the relationship between what you're showing, if any, what you're showing and what has been described back in the days as a visual imagery, so the ability to revive what we have seen before?
I think it's on anymore. Oh, it is. Yes, thanks. I think the answer is both yes and no. I mean, I think in terms of actual neural implementation, it might, at least the effects I'm showing in the visual regions, might be very similar to what goes on as a function of mental imagery. That said, for example, in this first study, when what I showed you with the dot, when you are repeatedly showing these sequences and then you just show the starting point, we show that effect, so those pre-play waves, they occur equally when you are attending those sequences and when you are totally not attending them. So when we give people a very demanding task at fixation, we still see those pre-play waves. So there is at least no conscious, or we believe that due to the fact that that task was very demanding, that there is no conscious sort of imagery of that sequence. And also in the last part, where we have this extremely rapid sequence of events, when we interview people later and ask them, like, did you notice any structure in your sequence? Almost all of the participants say that they are not aware of that. So that seems to be an implicit process, as has been often described for learning statistical regularities. So those things don't seem to require conscious access. And in that sense, to the extent that imagery is a conscious, effortful process, I don't think they can be explained by that.
More questions?
Hi. In one of the figures you showed, it looked like when you had the 4 dots, when you just showed the first dot, that was, it looked like it was weaker activation than it did when you showed all of them. Is that true? Is that what you found, or am I misconstruing?
Yeah, no, absolutely. Yeah, no, so anticipating, and that also makes a lot of sense, I think, because when you are actually presenting the dots, you get a strong bottom-up drive into all these 4, or into the neurons that represent all those 4 locations. Whereas in this situation where you just have the starting point, you only have the anticipatory and perhaps also a surprise signal, but not the bottom-up input. And we know that those top-down signals tend to be much weaker than bottom-up inputs. So in that sense, yes, definitely. Just anticipating something is associated with a markedly reduced activity than when you actually have the input.
So you're saying that explains even for the first dot that was shown?
Can you repeat that part?
So you're saying that it explains also for the first dot that was showing a weaker activity?
Oh, right. No, the first dot did not show weaker activity.
Okay.
I can quickly go back to that. Let me see if I can... right, yeah, I don't have the bars here, but yeah, I mean, there are some technicalities related to cortical magnification that may make it look like these dots elicit a higher activity than the ones over here, just because they are more peripheral. So there's less neurons devoted to the processing of more peripheral locations. But there was, in fact, no difference in activity at the location of the first dot for this condition where there was physical and this condition, as you would also indeed not expect.
Okay, thank you.
Yeah, you're welcome.
This is a fantastic experiment, this one in particular. I was really impressed. And I was just wondering whether there's a way to dissociate some effects of temporal attention versus temporal prediction here, because it's well known that in your retinotopic maps, when you're about to do, say, an eye movement, you're going to amplify the receptive field in the periphery, and this has been attributed to attention in the past. It's like a mix between spatial and temporal attention, but we can imagine that here what they're doing is that they're simply attending the next location in advance, and that would basically amplify that specific receptive field.
No, that's right. So we were worried about this as well, so that's why we had these 2 conditions manipulating the attentional state of the subject. I didn't have time to go into that part for this talk. So, but we had one condition where we actually wanted subjects to have attention on the dots, so they had to report whether the 4th dot occurred at the right moment in time or slightly delayed from when it was expected to happen. So then you really indeed have temporal attention first on the left, etc., moving along. But we also had a different condition where people had to, they always had to fixate, but at fixation there was like a rapid stream of letters, and they would have to press a button for like an X or and a Z and ignore all the other ones. And in that situation, attention was really disengaged from the periphery because there was a demanding task at fixation. And we find that there is no difference in amplitude of preplay between those 2 conditions. So that's why we think we ruled out the effect of sort of temporal attention here.
So my question is also about this experiment, but in the experiment you used just moving dots, a moving dot display, but the example you gave was a tennis match. Mm-hmm. So I'm just curious what you think about what might happen if you looked at the brain of the person who's acting. So a simple case would be Roger Federer just about to hit a stroke. Another case is when he's trying to hit the ball in the unexpected location, thinking that the other Nadal would be anticipating the expected location, but he's trying to hit in the other location. So do you have a sense of what might happen? Like, are we also replaying when we're trying to act on the world?
Yeah, that's an interesting question. I mean, obviously, I have no idea, but I would imagine that the kind of anticipatory mechanisms of how another person would potentially replay, or etc. That is definitely, I would think, a more higher order cognitive form of expectation than this kind of low-level expectation. So more akin to theory of mind potentially. So in that sense, I would not expect that those same mechanisms would also play out in the visual cortex. I mean, a wild hypothesis could be that the visual cortex of Roger Federer in that case would pre-play what is actually going to happen, whereas another sort of potential mental simulation happening higher up would be pre-playing what another person is think that is going to happen. But yeah, I don't know how I would be able to test that. But yeah.
Hello. You used the phrase the hallucinations, and I am curious about whether your results can say about something real hallucinations, how they work, about their mechanisms.
Yeah, again, it's not really my field, so I can't say anything with authority here. But it is known, if I'm correct, that the neurochemistry of schizophrenics, for example, is altered in the sense that, I believe NMDA receptors are sort of more, are more dominating. Or so I could imagine that in that case, so if there is this neurochemical alteration of balance, that would alter the strength of top-down signals compared to bottom-up signals, and that I suppose could lead to these kind of situations. So we know from neuroimaging work that there is activity, for example, in the auditory cortex when schizophrenics hallucinate. So it's probably very much related to what we see also when we see like visual activity when people don't hallucinate but actually imagine something. So, but if that would sort of invade the boundaries and would activate the whole cortical column, potentially due to this altered neurochemistry, I could imagine that would then lead to a situation where you would start perceiving your beliefs. Yeah.
This is actually about the same point, about the schizophrenia issue. Clinically, hallucinations in schizophrenia are actually very prominently verbal as opposed to visual. Not just verbal, but very often verbal.
Right.
So I wonder whether you could speculate a bit on what the connection is between hallucinations and language, actually.
Right, yeah, and I think the reason that it may be not so prominent in vision is also because we are very visual animals, right? It's a very dominant sense. We have a very accurate representation of the world in visual coordinates, much more so perhaps than in audition. So I think that might be one of the reasons why it's just harder to actually or for beliefs to become so powerful to actually override sensory inputs. Potentially, and that I think could be a difference between vision and audition. But then indeed, maybe also our thoughts tend to be more, indeed, like as you say, like linguistic, so verbal, therefore auditory rather than visual. So I think in that sense, a faulty sort of cognitive system would maybe more easily invade or pervade sensory representations in the auditory domain than in the visual domain. Yeah.
I think we have to move on. Thanks again.
Thank you.